Everybody can have a seat. Be fantastic. I'm Wayne Holden. I'm the President and Chief Executive Officer at RTI International. I want to thank everybody for coming today, including our online audience who has plugged into this. And we're also very thankful to have such a prestigious set of guests who are going to be on panels today discussing the issues. I also just want to note that we moved up the time on this to 10 o'clock, so there may be some late arrivals. I know we're getting a little packed in here, but we'll certainly try to accommodate as many people as we can for this very important discussion today. And I think as everybody knows who's in the audience today, Zika is a huge global health problem and a priority that is linked to severe birth defects and other types of health concerns. And there are leaders around the world who are struggling about what to do with respect to the Zika virus. And certainly leaders in the U.S. are trying to take a significant stance with respect to that. It's critical that we find out more answers about this particular virus and figure out what we can do to control what is now already a pandemic and could be a pandemic with very significant outcomes and complications for us going forward. We are involved in Central America and have an office there, and have been involved in a number of different activities in Central America that we really started several months ago based on some of our own resources to try to combat different issues associated with the Zika virus and the problems underlying it. What's been apparent to me is I've met with the steering group that we established back in January on a regular basis is that there are many, many more questions than there are answers about the Zika virus. And so we need a very comprehensive response going forward to try to address those issues and to be effective with respect to our public health response to this growing problem. We're pleased that there are so many federal agency representatives who are able to join us today. We're also very pleased to have university researchers with us, as well as researchers from the commercial sector here today, and some of our own researchers who will talk. And we're always pleased to have Congressman Price with us. And Congressman Price, I've known for many years since I've been here. He's always been a vibrant force in our community with respect to addressing issues, with respect to bringing issues to the forefront that need to be talked about, with respect to bringing the significant intellectual and research prowess of the Triangle to bear on these things. We've been very fortunate in this area to have a representative like Congressman Price, who I think is in his 14th term, 13th term, somewhere close. Yeah, that's right. Long term. But he's tirelessly advocated for many things on the Hill, and many of us have been involved in that over time and really appreciate his energy, enthusiasm, commitment, and insight into what's possible and bringing resources to bear on different things. And with that, I'd like to ask Congressman Price to come forward and give some remarks. Thank you.
Thank you, Dr. Holden, and welcome to all of you this morning. This is a wonderful turnout as we anticipated. Although this program's been put together on very short notice with the help of many, many people, there's just no question about the urgency and the salience of this topic, and so we're delighted to have you here, and we look forward to a productive morning. I want to welcome especially our guests from the research community from across the Triangle. You'll hear from them directly, and also our guests from the federal and state agencies that are involved in organizing and coordinating the research and the response. I want to thank my own staff, Nora Blaylock, Asher Hildebrand, Lawrence Klutz, others who have worked with the team here at RTI over these last couple of weeks to put this program together, and the results are what you see here today. People moving their schedules around, but eager to be part of this. And there's just no mistaking the urgency of what we're talking about here today and the importance, and that has been manifested in the response we've gotten as we tried to put this event together. As Dr. Holden intimated just now, it's not every congressional district in the U.S. where you could even think about having a conference like this. So it's a reminder once again of the richness, the diversity of the area we live in, the kind of research that's going on here, the kind of intellectual resources that we draw on in so many areas of national need. So I feel very fortunate to represent this area and to be able to be a catalyst in this case for a discussion that we all need to have. We have three great opportunities before us today. First, we have an opportunity to learn from some of our area's leading Zika researchers about the work they're doing to understand this virus and to design effective interventions to address it. Secondly, we have an opportunity to learn from senior officials about the government's response to Zika and the resources that are or should be available to help confront it. And finally, we have an opportunity to educate the public. So we do appreciate the media being here today, and we appreciate the potential of people who maybe aren't used to doing outreach all the time, but who need to do some outreach on this one in terms of public awareness in North Carolina, around the country, about the nature of this threat, the everyday steps that people can take to prepare for it. Given the limited time we have to realize these opportunities, I'm going to keep my opening remarks very, very brief. But I do want to say a word about the ongoing discussions in Washington about the federal government's response to Zika, which I've been involved in as a member of the House Appropriations Committee. To be sure, there's a lot we don't know. You'll hear a good bit of that this morning. A lot we don't know and need to know about the Zika virus, its causes, its epidemiology, its relationship to other conditions, the totality of its effects. But there are some things we do know. We know that Zika carries with it the potential for serious negative public health consequences, including microcephaly, which leave newborns burdened with horrible, lifelong physical and mental developmental defects and cost millions of dollars to treat. The estimate I've seen is $10 million average per case. We know that the CDC has warned us that the mosquitoes that carry this virus will reach as far north as Maine this summer, including the entire state of North Carolina, and that other countries and U.S. territories are already beginning to experience the impact of the crisis, with its full impact yet to come. And we know that confronting this threat will require a major mobilization of resources, public and private, federal, state, and local, scientific, technical, and social, that cannot and should not be impeded by the partisan divides to which we've become accustomed in Washington. And yet, more than 3 months since the administration requested an emergency supplemental appropriation from Congress to fund Zika research, preparedness, and response effort, I'm afraid this is exactly where we find ourselves. And there's no reason to sugarcoat it. Of all the emergencies and potential emergencies that Congress has been slow to address in recent months, from the Flint water crisis to the opioid epidemic, I think the Zika case, our inability to respond in a timely, serious, and sustained way, that's the most baffling one, the least comprehensible. I'm not going to dwell on the reasons for this failure. We're not here today to discuss congressional dysfunction. But I will let the facts speak for themselves. The administration has requested a total of $1.9 billion in emergency funding, including more than $800 million for the CDC, the Center for Disease Control, without requiring us to rescind spending elsewhere to pay for it. That's the normal thing we do in the case of a natural disaster or a pandemic. Senate Republicans, in a bipartisan basis, with Republican leadership, have approved a funding package of about $1.1 billion, including $450 billion for CDC. However, the House majority has just approved $622 million, a third of what the administration requested, including just $170 million for CDC. That's less than 21% of the request. Now, they stress this is only the fiscal 2016 portion, but by the same token, this creates great uncertainty as to the future availability of the funds. The majority of these funds would be cut from ongoing efforts to fight Ebola and other diseases. We simply have to do better than this. I'm not alone in this view. We can't afford to wait until cases begin spreading across the continental United States to get our act together. But if we're going to bridge a funding gap of this magnitude, Congress is going to have to hear loud and clear from the research and the public health communities, first of all, who know most about this crisis, but also from the American people, that any further delay is unacceptable. Now, fortunately, neither the researchers working to help us understand the virus nor the federal and state agencies leading the charge to confront it are waiting for the Congress to act. In my conversations with researchers, I've been impressed with how they're scrambling to move money around, to get the research going with whatever resources they can, to make do with the resources at their disposal. I think we'll all come away today impressed with the way these research organizations are improvising. The Department of HHS has already taken $500 million from ongoing Ebola funding. They've taken $50 million from money designated for the states and cities for public health preparedness. They've already moved that money around. But the question is, what comes next? North Carolina and the Southeast risk being at the center of this crisis. We're also uniquely positioned to be part of the solution. We have the brain power, we have the institutions here in the Triangle to make a huge difference. I think we're all going to appreciate this anew this morning. So we will hear first from the researchers, a diverse array of researchers involved in various aspects of this crisis, and then we'll hear from a panel of state and federal agencies who are attempting to coordinate the response. We appreciate everyone being here, and we look forward to an informative discussion. Thank you.
Good morning. If our first panelist could join me here on stage, that would be great. Great. Well, we've assembled quite a compelling session to start the day here, and we appreciate your attention to this important issue. You're going to hear from a number of leading researchers about this set of concerns. We've set aside traditional rivalries to bring together researchers from major universities in the Triangle, as well as industry and the nonprofit sector. And our discussion is going to be fairly wide-ranging, but there's a common thread here in that all the researchers that you're going to hear from are really at the frontier as we are addressing this virus. There's going to be brief opportunity for a question and answer at the conclusion of these remarks, and I'll offer instruction on asking questions at that time, but chances are that we're not going to be able to accommodate all questions that you might have. So we also are going to invite you to comment via social media using the hashtag RTI Forum and also the hashtag ZikaNC. I'm going to briefly introduce the whole panel to you, and then again we'll introduce each of our speakers before their individual remarks. We have with us today Sally Permar from Duke University, Michael Reskin from NC State University, Ray Taylor from BioCryst Pharmaceuticals, Aravinda Del Silva from UNC Chapel Hill, and Pia McDonald from RTI International. We're going to begin with remarks from Dr. Permar. Sally Permar is Associate Professor in Pediatrics, Immunology, Molecular Genetics, and Microbiology at the Duke University School of Medicine.
Thank you. So thank you again for putting this together, Congressman Price and all members of RTI. This is a great opportunity for us to start collaborations even, and like we said, that doesn't go on enough in the Triangle area, so we're happy that we get this opportunity. So I'm a pediatric infectious disease doctor as well as lead a research laboratory studying congenital infections. And so the one that I studied most frequently prior to Zika is congenital cytomegalovirus, which causes 40,000 infections in the US and 5,000 long-term deficits in children. estimated cost is $4 billion annually for that virus. So I would say we don't need another healthcare cost like that, which could come with Zika. We also have a vaccine for a congenitally acquired virus that has been extremely successful in eliminating a congenital syndrome, and that's the rubella vaccine. The rubella vaccine saves up to $7.2 billion annually. Seen in the graph is how it has eliminated congenital rubella infection in the Americas. So we need a Zika vaccine, and I work at the Human Vaccine Institute at Duke, and so we are very interested in furthering that work. I think there are three important things to a vaccine that we're going to need to overcome. One is understanding natural immunity and how that relates to the fetal infection. The other is having accurate diagnostics. We're not going to be able to tell if our vaccine works if we can't diagnose the infection. And then having animal models of disease that can accurately predict whether a vaccine is going to work. So I have previously worked in non-human primate models for congenital infections, including HIV and cytomegalovirus, and worked with David O'Connor at University of Wisconsin to help develop a congenital transmission model in non-human primates. And David O'Connor has been pushing the field forward by making his data available in non-human primates online in real time, and that has been a great asset to the community. And recently they showed that upon re-challenge, so monkeys can become infected, as seen by the viral load there on the first quarter of the graph. And then they later went back 70 days later and tried to re-challenge those animals, and the animals were protected. That's very good news for a vaccine, that natural immunity seems to be protective against a new infection. However, some potentially bad news that came from the monkey model is that while the viremia was pretty short-term in most of the animals they infected, between 7 to 10 days, and that accurately reflects what's going on in humans, pregnant monkeys, interestingly, had a much longer viremia. They were viremia up to 30 days for 2 animals and up to over 70 days for another animal. One animal had a more typical course. So this is potentially bad news for controlling this virus in pregnancy. This is either immune deficiency of pregnancy, that immune changes that happen to maintain the fetus, or potentially a fetal infection that's maintaining the maternal viremia. And so this needs to be studied and understood what is the missing part of the immune system that helps control this virus in pregnancy. So the work that we've been doing is trying to use the primate model to isolate potentially protective immunity. If we could understand what's the protective immunity in the primates, then we could try to induce that in humans. So here we've isolated the antibody-producing cells from those infected mothers and are starting to produce monoclonal antibodies so that we could try to see if infusion of those types of antibodies would protect against a new infection. There's other work going on at Duke, though, in additional animal models, particularly in rabbits. You can't do everything in non-human primates. They're very costly, and so small animal models are really important, and I think you'll hear more about mouse models that other universities are pursuing. But the rabbit model is potentially a good model because it's a good model of has a very similar placental structure and placental antibody transfer as humans, so that's one that we are pursuing. There's also very talented researchers working in the area of host restriction factors, host gene expression changes that happen upon infection with Zika, and importantly, the host gene factors that change upon neural stem cell infection, which has shown to be able to happen and likely the cause of the poor brain growth. Those studies are being led by Herman Statz, Debbie Silver, and Stacy Horner at Duke. Other important work is improving the diagnostics, as well as potentially making therapeutics or preventatives, particularly around monoclonal antibodies, and that's a particular strength of the Duke Human Vaccine Institute and Dr. Dee Gunn, who's here today. So monoclonal antibodies that recognize the Zika outer protein are very important, and they need to specifically recognize that protein. That's something that we are lacking in the diagnostics and is very important to overcome. And obviously a development of point-of-care testing, something that you don't have to wait several days to get your test back, is very important. In addition, these human monoclonal antibodies that neutralize Zika virus could be a therapeutic or preventative. Imagine giving a woman at the beginning of her pregnancy an immunization or a shot that would give her an antibody that would protect her through the 9 months of pregnancy. That would be an easy solution, potentially, before having an active vaccine product. And then we also have several vaccine candidates that are in production and testing. One is based on the yellow fever virus, 17D, so there's an effective vaccine for a very similar virus, yellow fever. And so using the backbone of that virus to insert the Zika outer envelope proteins would be a good approach because the safety profile should be similar to an already approved vaccine. We're also working towards a modified vaccinia ankara, or MVA, vectored Zika vaccine that expresses the outer proteins. This is also a very safe vaccine strategy that's been used for a number of other candidates. And Duke has also been building up GMP facilities to facilitate production of this proteins and vaccines in order to do human testing in small numbers at least. And then the other really important part is to try to understand natural immunity. And so far, the data that we have is not every woman who becomes newly infected with Zika virus will pass it on to her infant. And moreover, not every woman who becomes infected with Zika virus will have an affected fetus. And so we have to understand the differences there, because that will help us drive our vaccine development. And so we've been working with a clinical site in Brazil through the Duke Global Health Institute to try to obtain new patients who are newly infected with Zika and have confirmed infection, particularly those in pregnancy, to be able to study what their immune response was before the infection, during the infection, and how that relates to their fetal outcome. And these types of studies are important, but not only at a single site, they need to be multi-site in order to really understand and get enough numbers to make these conclusions. And so that's something important to fund for the future. And also I want to make a plug for the regional biocontainment laboratory that's available at Duke. This is one of just a few federally funded centers throughout the US that has BioLevel 3 capabilities that can also give added safety components to work with Zika virus. Charlie McGee is here. He is one that has worked to develop Zika resources for the community, including virus stocks, neutralization assays, and the ability for animal model testing to go on there as well. And the contacts are Greg Simpowski and Charles McGee, who's here. So thank you very much.
Great, thank you, Sally. We really appreciate it. We're now going to hear from Michael Reskin, who is an assistant professor of entomology at North Carolina State University.
Can everybody hear me okay? I took a slightly different perspective on this, which is the mosquito perspective, but I also wrote this brief little talk to kind of educate everybody, because I feel like there's a lot not known about by the general public and by even the scientific public about the mosquitoes involved here. So first, just a little overview of Zika epidemiology. Essentially, I don't know what percentage, I don't know if anybody does, but probably 95 to 99% of current cases of Zika in the world are transmitted by mosquitoes. That being said, there is sexual transmission of Zika. And I would venture to say, although maybe some of the public health folks know something I don't, but 100% of cases acquired in the United States have been sexual or mother to child transmitted. So this is something different than we've seen in probably any similar mosquito-borne virus. That being said, because most cases are going to be transmitted by mosquitoes, at least in the developing world, understanding the ecology of the mosquitoes provides assessment of risk and also our most fruitful avenue for prevention of disease. So again, a little bit of biology. We have two major likely Zika vectors in the United States. Okay, on my left, your left is Aedes aegypti. I'm looking at it here. Anyway, which is probably the primary vector. It is pantropical, urban, and domestic. It is essentially our pet mosquito, if you will. Very similarly, ecologically, is Aedes albopictus. This is the Asian tiger mosquito. It is pantropical, but also temperate. And in fact, when the CDC says a Zika vector could be found in Maine, it's this mosquito that they're talking about. It is generally more suburban, although it certainly can be found in urban areas. And I would say more peridomestic than domestic. In the tropics, in the developing world, you can find Aedes aegypti inside people's houses, often, breeding inside people's houses. Aedes albopictus, maybe, but also outside of the house. Okay. So they share a lot of characteristics. They're pestiferous, they live close to people, and they bite people often. They are a low-density mosquito. They're well spread out. Although the flight range of individuals is probably somewhat limited, that may have a lot to do with the density of people around, we generally can find them anywhere in a suburban neighborhood, at least Aedes albopictus in Raleigh. Or in Miami, we find aegypti in almost any neighborhood. But they are low density, and this makes certain traditional mosquito control approach is difficult to implement. Okay? And they prefer to use containers to lay their eggs and have their larvae develop. And this can be artificial, like your coffee cup that gets thrown out, can fill with rainwater, or natural, like tree holes. These also present difficulties for control. There's no clear target to hit. Something I should add about this as well, these are generally diurnal mosquitoes, meaning that they bite during the day. So your bed net, not really effective for these mosquitoes. Okay. There are some differences, though, particularly in distribution in the United States. And so this figure on the upper left there shows the distribution of Aedes aegypti in yellow and Aedes albopictus in blue over several years, and these are data generated from my lab. Along an urban to rural gradient in South Florida. Okay, and so you can see in 06 and 07, aegypti was essentially restricted to the urban coast. This is the east side of Florida. And actually, we saw a spread of aegypti in that intervening 6 years between 07 and 2013 as well. There are also some other differences between the two. Aegypti, well, albopictus continues to spread. It's well established in North Carolina, so we won't see spread in North Carolina. It's here. But it continues to spread further north. So right now, the invasion front of Aedes albopictus, the Asian tiger mosquito, is essentially New York, southern New York, but it will continue to probably spread. That being said, Aedes aegypti seems to be making a comeback. I didn't tell you kind of the whole story, but Aedes aegypti used to be more abundant in southeastern United States. When Aedes albopictus invaded, Aedes aegypti went locally extinct in places, including North Carolina. But we're seeing this resurgence of aegypti. And then, probably most importantly, is Aedes aegypti is generally considered the more critical vector. It is more faithful to feeding on humans. It lives longer, and those data to the right show that it lives on average about 15 days longer in laboratory studies. That's a laboratory study from my lab. And this is critical for its role as a vector. Long-lived mosquitoes transmit more disease. And also, Aedes aegypti is more closely associated with people and living near or in the home than Aedes albopictus. Okay. So there are several traditional approaches to controlling these mosquitoes, and none of which are all that great, frankly. Probably the most basic control approach, and something that was implemented as soon as we found out these mosquitoes transmitted disease, is cultural control. That is, cleanup campaigns. Get rid of the larval habitats like the tire or the rock pool that are not being maintained, or attack those larval habitats in some way. The second approach is the chemical approach, and this came to increase prominence after World War II, and we had effective chemical pesticides. This can include area-wide spraying, so fumigation of area or ultra-low volume spraying. However, for these mosquitoes, because they're low density, because they're day biting, because their behavior doesn't make it easy to hit them with large spray campaigns, they're not that effective. What seems to be more effective is your household approach. And we've seen this with the rise of backyard spray control. I'm sure all of you have seen a sign in your neighborhood that says mosquito something. I won't say any company name for fear of that. Recently, and this is something that people have known about for a while, but recently these have been increasingly implemented effectively, are autocidal gravid ovitraps, and that's a bit of a mouthful, but AGOs. And this actually attacks the egg-laying female mosquito by attracting them to an artificial habitat that kills them directly, either through chemical means or sometimes through sticking to something so they can't. And this is actually an important approach because it can effectively target the mosquitoes most likely to transmit disease. And then finally, not implemented in the United States, are genetically modified organism approaches, or Wolbachia, which is an internal intracellular bacteria found in mosquitoes. And this gets pretty complicated. We can talk about it another time. Or other male-based control, even dusting males, and the males are the ones with the frilly antenna on the right there, dusting males with an insecticide. And when they go to mate with females, it kills the females, or kills the larvae. And these are currently, some of these approaches are currently in use in Brazil, Panama, Indonesia, a few other places around the world. They tried to implement a program using genetically modified mosquitoes in the Keys, but public outcry was such that they did not implement that. So this has not been done in the United States.
That's no
Oh, okay.
We've just been through FDA approval. They're just about to.
Okay. All right. So previously they tried to do it, but apparently this is from Oxytec's group. Yeah. Okay. So apparently are and Oxytec. Well, I won't be a shill for them either, but they've had a lot of success in Brazil and other places around the world. So, okay. So talking about risks from the mosquito-borne perspective, there's already sexual transmission of Zika in the United States. And there's already a case just this week of a woman giving birth to, I think, a microcephalic baby in New York, but that was an imported case. So a study that I was a small part of came out recently and modeled the risk of mosquito-borne transmission in the United States based on travelers coming to the United States, season, which is those pie charts, and then the presence of Albopictus or Aedes aegypti as vectors. So outside of summer, we probably don't have much risk of mosquito-borne transmission in the United States because there's not mosquitoes outside of summer and into the fall, I would say. There's also an effect of latitude. Okay, as you go north, you're just less likely to get a sustained mosquito-borne transmission of disease. And this interacts with season, of course. It seems kind of obvious if you think about it. I think that urbanization can be another risk. As we saw in South Florida, Aegypti is dominant in urban areas. Aegypti is likely to be a better vector for Zika. But there's no reason to think that we won't start to see Aegypti populations established in urban centers in Georgia, North Carolina. We already see it in New Orleans, Texas, et cetera. Okay. And then lifestyles are critical as well. We generally don't see large outbreaks of mosquito-borne, especially these, dengue, chikungunya, or Zika, which are going to have similar epidemiology because they share vectors. We don't see large outbreaks in large part because we spend a lot of times inside. We don't tolerate mosquito bites like you might if you lived in an impoverished community in Guatemala. It's very different in that way, too. Okay, so my final part is about surveillance. I think that mosquito-based surveillance is going to be an integral part of any Zika response, and it can direct public health efforts in an efficient manner. It's often poorly invested in until an outbreak is upon us, like we have now. It would have been much easier, I think everybody who's in public health right now, if every county had an established mosquito control district in it in North Carolina. That is simply not the case, and it leads to lots of difficulties in implementing surveillance. And I just want to note that the CDC has now requested that even travel-associated cases have some kind of response, including surveillance and maybe even control. And this, for the states and for local municipalities or local districts, this could be a very big barrier. Okay. So thank you, everybody, for your time.
Thank you, Michael. Next, we have Ray Taylor, who's Vice President of Program Management and Project Leader at BioCryst Pharmaceuticals.
Yeah, hi, good morning. Thank you for RTI for organizing this and getting that very flattering picture of me. I don't know where you got that. I'm sure it's social media someplace. And thank you, Congressman Price, for your support here and activities of research and development. I'd also like to thank our panels, as well as our colleagues from HHS, who I've worked with in the past for many, many years, and their leadership is really important here. So I get a chance today to talk about potential therapeutic, which I'll go into a little bit more detail, a promising drug that we've been working on, primarily with funding from the U.S. government. It's a broad-spectrum antiviral. The compound name is BCX4430. Sometimes we refer to it as 4430. So if I slip and say 4430, it's BCX. It's one of our compounds that we discovered out of Birmingham, Alabama. Just briefly, our company, if you don't know us, we're a very small company, about 60, 65 people, right down the road here. So it took me about 5 minutes to get here this morning. Our headquarters has the clinical development and medical research, as well as our regulatory and our toxicology and our staff functions. In Birmingham, Alabama is where our research is. We were spin off at the University of Alabama, Birmingham in 1986. We also have our pharmaceutical development in Birmingham as well, too. So a little bit about BCX4430 for those who don't know about it. It's a adenosine nucleoside analog, and that's a lot of words there, but basically it's a direct-acting antiviral that converts to what's called the triphosphate. That's the diagram up in the upper right. It does it very quickly. That's the active part of the drug. It gets incorporated into the virus replication, the RNA, and stops the replication, so that the virus replication, just like most antivirals, gets stopped, and then the immune system takes over after that. We have a broad spectrum in which we've demonstrated efficacy in a number of animal models in Ebola, in Marburg, and yellow fever, as well as some others. And I'll show you some data that we generated on Zika, which is why we're talking today. So where are we with the development program? Well, I'm happy to say that we've just completed dosing of our phase 1 clinical safety study of about 80 or 90 subjects. We have an IM formulations that we've been looking at. It's generally safe and well tolerated, no adverse events to report except for some injection site pain associated with the injection. We will be finalizing the data, which is coming in, I think, this week actually as I speak, and we'll probably have a publication on that. We've also conducted non-human primate efficacy studies. The slide is wrong. I apologize. I must have been sleepy last week. We've actually done 3 studies in Ebola, in cynomolgus monkeys and rhesus monkeys, showing protection of survival models, as well as an early Marburg study. Unfortunately, the Ebola is not published, but the Marburg is, so that was in a Nature journal. And we showed survival benefit in both those models. The Marburg model was a delay in which treatment was initiated after 2 days, I think, delay after a viral challenge. The Ebola, those weren't, but we continued to do studies, and they're all done at Fort Detrick under U.S. AMRID. Always an issue is drug supply, or with these new research compounds, can you make the drug? I'm happy to say that our first delivery of GMP material for our clinical trial was in November 2014. We continue to make additional drug supply in our manufacturing facilities in Wisconsin. We're at a kilogram scale process, but basically what that means is that we can start making enough, and we are making enough to supply clinical trials as well as potential studies we might do in infected areas of the world. And then lastly, one of our major focuses now is our IV program. We know that an IM would be useful, but delivering IV might also be needed, and particularly in hospitals where patients are sick or getting fluid. Remember, this was originally focused on Ebola and the outbreak there. So the IV program is a little bit behind the IM program, but we hope to be advancing in the clinical studies either later this year or early next year. And I did forget to mention the highlight box at the bottom. We've had federal funding from a NIAID contract since 2013. We also got a BARDA contract last year as well, and those are still active that are taking us through advanced development. So then a little bit about Zika. As I mentioned, our original focus for this was Ebola, and the funding for this project is really from the Ebola funding that was requested by the President to respond to the crisis. Late last year, we thought about Zika as a potential virus that we should study. We had activity in other viruses in the same family, the flavivirus family, including Rift Valley, West Nile, and yellow fever. So we contacted one of our researchers, Dr. Justin Juliander, out in Utah State University, and under a preclinical services agreement with NIAID, we contacted him about any research he was doing with Zika. That was late last year. We advanced to the beginning of this year. He has and there are these models around, an immune deficient mouse model, which when the mice are infected, it's a survival model and they die. And of course, that's not what happens with Zika, but it's a way to test your drug to see if it has antiviral effect. What you see on this slide is that this is the first of 2 studies. I'll explain both studies in just a minute. In this study, 2 doses of 4430 were administered to the mice prior to inoculation of the virus of about 4 hours. And then comparators were vehicle or placebo, as well as ribavirin. You can see the curves there, the Kaplan-Meier curves there, in which the highest dose was I think one mouse died, but it was protective. Even at half the dose that we did, 300 mg per kg there for 8 days, was also statistically significant versus ribavirin and vehicle. You can also see that on the right-hand side at day 5, the viral load. Again, you can see that 4430 was able to reduce the viral load associated with the infection. Then what we did, what Dr. Juliander did, was he re-challenged them. So the surviving mice that were infected with the virus and then were administered 4430 were re-challenged at day 28. And what you see here is, again, They had mounted some immune system, even though they had a interferon deficiency. They had antibodies that were developed, and all but one survived the re-challenge. So something was generated to allow them to be resistant to the virus and fight back. And then you can see the controls. Those were ones who weren't originally infected, then were infected, and then they all succumbed to, as you would expect. So that was all nice and good. But then the next question is, can you delay treatment? So this, remember, the first study was upon a virus challenge getting drug basically at the same time. So the next study we did with Dr. Juliana was delay treatment. Treatment was delayed 1, 3, 5, and 7 days. And as we had hoped and expect, you see how the antiviral, the longer it takes to treat, the poorer the outcomes. However, as you can see, there's a statistical significant survival for every single one of the groups versus placebo. So particularly with the ones that were treated very quickly. So this is as far as we've gone with this particular drug in investigating Zika. This is my last slide, but I'll just summarize very quickly. We are now investigating doing studies in non-human primates. We've talked with some of the groups that have been mentioned here, as well as understanding the pregnancy part of that. That would be the next step as well, too. And then lastly, one of the things that will be very interesting here is figuring out what the exact optimized dose is. This dosing, again, was for 8 days. We believe we might be able to do better than that and actually have a loading dose first, followed by either a maintenance smaller dose or maybe no dose at all. But that's a quick summary.
Thank you very much, Ray. And now we'll hear remarks from Aravinda Del Silva. He is a professor in the Department of Microbiology and Immunology at the University of North Carolina at Chapel Hill.
Thank you, Dr. Holden, for organizing this, and Congressman Price for really being the motivating force behind getting this group together. One thing I really want to emphasize is here in the research triangle, this tremendous expertise. We've talked about, local expertise, maybe issues that have to do with dealing with the national prevention of arboviral infections, but really the groups that are working here in North Carolina in this region are really, the work has impact way beyond the US. It's really the work that has an impact on really dealing at a global level with emerging arboviruses. So what I want to do today is not really get into some of the data, because there's over 10 groups at Chapel Hill working on Zika, and in 5 minutes I couldn't really do justice to some of the work that the different groups are doing, but to really give this community an overview of the kinds of research activities that are taking place at Chapel Hill, and really looking forward to collaborating and engaging with others with similar interests. Now, one thing I want to emphasize is, there's things about Zika that are very different and unexpected. But there's also many things about Zika that Zika is a flavivirus, and we know a lot about these viruses. And so as we think about dealing with Zika, a lot of the things that we can do are actually going to be based on knowledge we already have based on other flaviviruses, not to try to reinvent everything. Okay, there's some things that are very similar between Zika and other flaviviruses like dengue. And then there are also aspects that are quite different. So Zika suddenly showed up a few years ago in the Western Hemisphere. But this is a trend that we've seen over the last 3 to 4 centuries. Okay, these mosquitoes, Aedes aegypti, which originally is an African native species, these viruses that are maintained in non-human primate cycles in parts of Africa, and the fact that these viruses are moving out and moving into other parts of the world. So yellow fever, which is transmitted by the same mosquito as Zika, was introduced into the Americas in the 17th century. But that pattern continued. Dengue was introduced in the early 1900s, and the first epidemics of dengue hemorrhagic fever were only seen in the Americas in 1982, so relatively recently. Chikungunya was first introduced to the Americas in 2013, and now Zika. And all of these viruses have the same urban ecology. Okay, so in that respect, it's not surprising that Zika emerged. Okay, it's humans are the major vertebrate reservoir, and Aedes aegypti, the mosquito vector. And so I think we really need to take what we've learned from all these other viruses in terms of the epidemiology and transmission and think about controlling Zika. So people ask, why don't we just control mosquitoes? Well, we've been trying, before Zika came along, we were trying to control mosquitoes, and there's already a tremendous amount of work. Okay, so to build on that work, There are also things that are really unusual, right? There's no precedent, and that's why I think we are all here, is the transplacental transmission and the birth defects. That's unprecedented, and so is the sexual transmission. And those are really new, unexpected findings that need to be studied. At Chapel Hill, we really have a long history of studying arboviruses, going back for decades, and so there's real expertise. So my group and others, we've been studying dengue viruses for many years. So dengue is very closely related to Zika, but distinct. And we have a program that looks at all the way from epidemiology, human immunology, and pathogenesis, including a lot of international work, both in South Asia as well as in the Americas. More recently, we've heard about the importance of vaccines for dealing with Zika. There's a tremendous amount of work right now going on dengue vaccines. Okay? 3 vaccines are right now in phase 3 clinical trials. Okay? And our group at UNC, we've been working very closely with all the leading vaccine developers, because we've done a lot of work on how after natural infection dengue viruses are cleared, and this information has become critical for evaluating the leading vaccines. So we're working in close partnership with all the leading vaccine developers. And as you can imagine, as these clinical trials are going on, for example, in South America, Zika is sort of adding another layer of complexity to understanding and interpreting these vaccine trials. Okay? The second area with respect to dengue vaccines is we also have a program where not supporting the leading vaccine developers, but also developing our own vaccines. This is a program with collaboration with Liquidia Technologies here in the Triangle, using nanoparticles for delivering dengue and influenza vaccines. And this is a collaboration with our colleagues at Duke as well. And when you think about the first 2 points here are about dengue, but there's also a lot of expertise in alpha viruses, okay? Another closely related family of viruses that are spread by mosquitoes. And there's a lot of expertise here at UNC on Venezuelan equine encephalitis, Ross River, as well as chikungunya, which is spread by the same mosquito as the Zika. These studies are supported by both the federal government as well as private foundations. So I just have a couple of slides about some of the key outstanding questions that I see in Zika, and some of these have already been mentioned in the work that's ongoing at Chapel Hill. And a critical issue is diagnostics. Okay, so all these reports you see about so many cases of microcephaly, so many people being hospitalized for Zika, many of these are just a clinical diagnosis. Okay, we still don't have the laboratory assays in place for accurately detecting Zika infection. This is a huge problem. 75% of the people who are infected don't show any symptoms. So if you're only looking at the tiny fraction of people who get very sick, you're really unable to track the epidemic. Okay? So we need diagnostic assays. Another complication is Zika is being transmitted in areas where there's already a lot of other flavivirus transmission. And without getting into details, having a previous flavivirus infection really complicates the diagnostic. So we really need to come up with simple diagnostic. The good news is, while the problem is complex, this is highly solvable. We know enough now about how we can go about coming up with simple, specific diagnostic tests for Zika. Okay? And so this is an area that we are working on at Chapel Hill. We are working closely with both the local state health department as well. We've already met with them to support their efforts here in enrolling the state health lab, as well as the CDC, given the expertise we have on flavivirus diagnostics, to help the group in CDC work out some of the diagnostic problems. And I've listed some of the different groups that are working on the diagnostic issues. And I also want to emphasize that while we need more funding to sustain these efforts, it's been remarkable how responsive the NIH has been in terms of, at very short notice, adding supplements to our existing grants to support these initial studies. The second area is prevention. I think when we think about vaccines, the good news is that there are already excellent vaccines against this class of viruses. So we're not dealing with trying to develop a vaccine against HIV, malaria, or TB, where there are really fundamental problems, barriers to be overcome. For flaviviruses, people naturally, after they're infected, make excellent responses that clear them. There's already vaccines against yellow fever, Japanese encephalitis. The first dengue vaccine was just approved for use in certain countries. So the promise is excellent. The prospects are good for Zika vaccine. Some of the special challenges are, usually we think of pediatric vaccines, for example, dengue. In the case of Zika, we have to think about trying to vaccinate people either early in pregnancy or people who are maybe trying to get pregnant, and there are some additional complications where some of the live virus vaccines that have been developed that are proven to be most effective are contraindicated in populations who are in people who are pregnant. And again, so a lot of, through supplemental support we've gotten from NIH, we're now exploring the nanoparticle platform that we've been developing for dengue as a way of developing Zika vaccines as well, because that's a non-replicating, non-live vaccine, which could be used in populations where you couldn't use a live vaccine. And finally, this is, I think, the area of the pathogenesis. How does this virus cause disease? This is where I think where Zika really stands out and is so different from these other viruses and other flaviviruses. And I've listed some of the key questions I think many of us are interested in. There's already both cell culture and animal model studies led by Helen Lazier, who's in the audience here, a new faculty member at UNC. And I also really want to emphasize the importance of global health and support for doing international studies. We can learn a lot from animal studies, but if there's one thing that we've learned from these flaviviruses is the importance of doing human studies, because some of the details actually that you see in the animal models are not seen in human disease, and often some of the critical observations in human disease cannot be reproduced in the animal models. So it's critical that we don't just see this as something where we do domestic work, that we invest in doing these global health collaborations to really study this virus in the setting where it's been actively transmitted. So I'll stop there and thank you.
Thank you very much, Arvinda. We're now going to turn to Pia McDonald, but just before we do that, just a brief word. Right after Pia's remarks, we will have an opportunity for a few comments and questions. And I'm going to encourage those of you that do have questions that you're formulating to be prepared to advance up to the stage. We will have a couple of folks who will be available with a microphone, but will require you to get out of your seat and to come up. So you can be thinking about that during Pia's remarks. Okay, in addition to Pia's remarks, which are very important. So Pia McDonald from RTI International. Pia is Senior Director of Applied Public Health Research here at RTI. Pia.
Thank you, Brian. So I'm here today to talk about the ways that RTI is addressing the Zika virus and ways we are engaging in the research. So first I'll talk about our internal research and development work, and then I'll talk about our externally funded research, as well as research gaps. But my panel members have done a fantastic job, so I'll go very quickly over that bit. So for our internally funded projects, we're looking at the use of drones to support vector control. So we're using drones, and we've done flyovers on 4 villages in Guatemala, and then coded that data for possible breeding sites. We've then downloaded that data onto tablet computers and given them to the vector control teams on the ground to allow them to use those during their spraying and other eradication efforts. We're also looking at the use of smartphones to collect data and to support families who have microencephalitic children in Brazil. So smartphones can be used not only for primary data collection with the families in real time, but also used as a way to push out information to the families and intervention strategies for them as they raise their children. We're also studying the potential for aerosol transmission of the Zika virus. We know that the virus can be found in human saliva, but what we don't know if the saliva is aerosolized, can it be transmitted to humans? We're also monitoring social media and the public's response to that social media in an effort to have that information when we design effective communication campaigns. So we're monitoring media in Guatemala as well as in the United States. We're looking and studying the costs associated with various vector control programs in the United States as a way to better figure out which ones we should be using in a cost-effective way. We are designing an adult prospective cohort and biorepository in Guatemala so that we can study Zika virus for the longer term. Our externally funded projects, primarily through NIH as well as the CDC Foundation, we're the data coordinating center for the international cohort study of Zika in infants and pregnancy, or otherwise known as the ZIP study. And you'll be hearing more about that today from our NIH colleagues who are here visiting. We are also the statistical and data coordination support for blood transfusion transmission studies through the NIH REDS program. Through the CDC Foundation and with them, we're working on a Zika virus prevention campaign in Puerto Rico. I'm not going to dwell very long on the various gaps in Zika research because everyone's done a fantastic job of addressing those. But I did want to just highlight the areas that RTI is poised right now to address. So for example, around clinical and epidemiologic studies, right now, as we've been hearing about, there are many different clinical studies going on internationally in various countries, as well as epidemiologic studies. RTI is very interested in making sure that these data can be compared, that they can be combined and integrated such that we can answer the many questions around Zika virus. So if everyone's collecting them and not coordinating, we're worried that we won't get as robust results and findings. These are some of the areas. And in summary, as you've heard, we've got a fantastic group of people here assembled today, as well as very esteemed colleagues that represent different research institutions and organizations and companies. And together, we really can make a difference in solving the many unanswered questions. And there's many more questions than there are answers today that we look forward to working on. And with that, I'm going to hand it back over to Dr. Southwell.
Thank you very much. It's been a great group of initial remarks and a good start to our day. We are going to turn attention now to questions that you may have. And so I'm going to invite anybody that does have a question to approach here, and then we will go around and take as many questions as we can within the 5 minutes that we have. I am also going to take a question first from our press colleagues in the back. If you could go ahead and address your question to any of our panelists, that would be great.
Sure. I'm Steve Gilliam with the Raleigh News and Observer. This question is probably best for Dr. De Silva or Dr. Reiskind. How would we in Raleigh know that isolated Zika cases have become an epidemic?
Could you repeat the question, actually? I'm sorry.
How would we in Raleigh or the Research Triangle area know that isolated Zika cases have gone from just being these isolated cases to an epidemic?
Well, I mean, and again, there are probably public health folks who are on the front line here, but if there's no travel history and they have a positive diagnosis of Zika, a single case would be huge because it would suggest that the transmission was local. Then you might ask about sexual behavior, sexual partners. And if those are all negative for travel history, then at least as a mosquito person, you would start to assume it was mosquito-borne, and then you would have to have a very robust public health response.
Yeah, so let me just add another comment to that. So, we've seen the maps of the distribution of the mosquito vectors, okay? It doesn't mean that wherever the mosquito is, there's a risk of there being local epidemics of Zika, right? So in order for an epidemic to happen, you have to have the mosquito, but you also have to have enough people being bitten by the mosquito so that the only way the mosquito acquires infection is by biting a person. So the mosquitoes don't just transmit it within the mosquito population. So, right? So there has to be enough contact between mosquitoes and humans in order for local transmission to be sustained. And right now, so I think this is where we can take a lesson from dengue, right? Has there been, because the ecology of dengue and Zika are similar, and we've had dengue now for decades, dengue hasn't taken off in different parts of the US except for some outbreaks in the Florida Keys, where there's probably high enough a density of mosquitoes. There's evidence in South Texas for just local transmission, sustained local transmission for a few years. So it's those areas where we should be looking out for maybe small local epidemics. That will also probably die out as we head into winter. But I think at this point, we shouldn't be worried about the virus coming and just suddenly causing an explosive epidemics in North Carolina.
Great, thank you very much. So we'll take a question here from each side of the room. Brianne, and if you could announce your name and affiliation.
My name is Ramabhadran. I'm an independent consultant working on global health issues. The question, I have several linked questions basically. One is, what is the serology I mean, I guess you detect it by serology. So what's the serology in men versus women versus pregnant women? And secondly, what's the diagnostic criteria? Somebody walks in a clinic, how do you find out it's Zika? And then the question to BioCrest is, how would you apply this if the diagnostic is not there?
So I could start just as a clinician, seeing, for me, children whom we may be investigating them for a congenital infection. We would start with the first contacting our state lab if we had a question about a patient that may have been affected by Zika, a travel-related case, and we would consult with them about the most appropriate diagnostics. Right now, the PCR test is very specific, and so that would be the one that we would rely on for specificity. And then there are serology tests that at this point are less specific and are cross-reactive with other flaviviruses. And so that's what needs to be solved. But I think we'd really work closely with the health department to get the most ideal testing. The other thing that I'll say that may provide longer ability to test for the virus itself by PCR is you can detect the virus in saliva and urine. And so those tests may become more available for longer-term diagnosis of the virus itself and not just the serologic response.
So you said if you had to wait for the child, how do you do that in adults?
So I was speaking just for my pediatrics expert clinic, but it would be similar in adults as well. I don't know if anyone else wants to address.
Yeah, maybe to your point, I think the rapid diagnostic is a really important thing. I think it was already mentioned that that's very critical, like with any of these, like influenza, for example. It's the same thing. I think the application of an antiviral treating at time of infection is very important. But we're seeing some of the data about persistent viral shedding. There might be opportunity there in which you see it in saliva and semen and maybe even in sexual transmission where males are still shedding virus, that might be an opportunity for using it there. But I think until you have rapid diagnosis, because the viral load goes very quickly, some of it's asymptomatic too, so treatment window is very, very short in that situation.
Great, and then actually we'll move on. What we want to encourage is a discussion after as well. That'll be great. And so I know many of our panelists have agreed to stay around for a bit of time after, and we may be able to have additional discussion. We'll have time for 2 more questions, and then we'll have to wrap up and move on to the next session. So, Laura, if you would like to offer that.
Hi, my name is Solzhen. I'm from Duke University, and I worked on pheronomic effect of Ebola and looking at the role of private sector in Ebola response in my research. So I have 2 quick questions, if that's okay, Brian? I have 2 very quick questions. My first question is for Michael, and this is regarding, I'm very curious whether you've seen a difference in terms of both Aedes albopictus and Aedes aegypti. Is there a difference in the way how they transmit the virus? Or, have you found Aedes albopictus viruses that are carrying Zika virus? So that's my first question. My next question is more of a comment/question. So, obviously looking communication, and PR mentioned a few comments on the importance of behavioral sciences and then the importance of implementation. And I feel like there's a lot that we can learn from Ebola response, both in terms of how the misinformation impacted the control of the disease, and also with regards to funding and involving private sector more. So I'd like you to maybe speak a little bit more on what RTI is doing on that. Thank you.
So very quickly, in a number of laboratory challenge studies, Aedes albopictus has been shown to be an effective, competent vector for Zika. And then about 2 weeks ago, they found a Zika-positive albopictus in Mexico. However, the role of albopictus, because there's plenty of albopictus in Brazil, remains unclear. It still seems likely that Aegypti is the primary vector for Zika.
Okay, so the question is, what are we doing sort of that's borrowed from the Ebola response and the misinformation there that happened? And as on my last slide there with the questions, we are working on trying to figure out what are effective public-private partnerships to engage on these communication campaigns. And it's actually Dr. Southwell's group who's working on that and looking at, can we partner ministries of health and private sector groups to create better campaigns. And I don't know if, Brian, you want to make any comments on sort of the in-depth part of that?
Well, in the interest of time, I'll just note that it is an exciting opportunity for us to look at, and it certainly is going to be a piece of the puzzle, but it's not going to solve the issue on its own. And that we're very cognizant of the fact that behavior is going to be part of the equation, but there are many other factors here, and we're going to be looking at those. We'll have time for these additional 2 questions. Yeah.
I had a question for Dr. Permar and Mr. Taylor. You both have compounds or virus, various compounds in the line for to treat this. What would be the next step in a strategic manufacturing platform to bring these products to wide availability?
I think I can talk mostly from the monoclonal antibody side, which is what we're pursuing at Duke and more than one group at Duke pursuing. And I think that would be first showing the in vitro activity, so against the virus itself in culture, then showing some activity in animal models in either preventing infection or reducing viremia. And then GMP manufacturing is a big component there. And so it would be ramping up for that, whether with a commercial partner or, again, there's some small GMP capacity being developed at Duke as well. So that's kind of the stages, but obviously involving commercial partners and government partners at an early stage is really important.
Yeah, so for 4430, as I mentioned, the GMP manufacturing is already taken care of. We're ramping up. We have funding from BARDA to continue to scale that up to even higher amounts. I think for 4430 as an antiviral, I think the key is, you mentioned to get it to a product to be approved, is what are we really treating and how are we treating it, I think is the most important thing. We have some interesting, promising data in a mouse model, but until you get to higher species or even maybe even some studies in infected unpregnant women would be needed to see if the antiviral effect is there. So I think how it's going to be used has to be defined. And there's going to be additional research that has to do that. I'm interested in seeing the data that David O'Connor group is having with the pregnancy. Viral shedding is coming later. I don't know if there's a role there. But I also think for us, if pregnant women is an area that we need to focus on, formal reproductive toxicology work needs to be conducted. We do intend to start that work again under our BARDA contract in August. So that would have to be done. We really have to understand the toxicity profile of this compound, but we also have to understand what's the right dose. So we've been studying it mostly in Ebola, very different disease than Zika, and we're understanding what that dose looks like. The dosing regimen may be very different for Zika, so that work has to get done. That would primarily, in my opinion, be done in a non-human primate model, and then we could progress that to humans.
Okay, we'll turn to one final comment and question here, and then also if the panelists have any questions for each other, we would have time for 1 or 2 of those.
Hi, I'm Dr. Derek Nimmo from Oxitec Limited. Some fantastic research going on here. This is my first time to North Carolina, and you guys are doing some fantastic stuff, and we're really desperate for a Zika vaccine. How long, and this is a horrible question, but how long do you think we're going to have something on the table that can be used to fight this disease?
Okay, I'll take a stab at that. So, I think the time it takes to develop a vaccine, it's one thing to show that you have an effective vaccine, say, in animal studies, but it's all the regulatory processes and the clinical trials. Those take years. To have a vaccine that's approved in 5 years would be very fast. Now, I think with Ebola, there may be some new models for fast-tracking vaccines, but sort of I don't have the expertise in sort of the regulatory landscape for fast-tracking vaccines. But scientifically, conceptually, I think a vaccine is not that far away. The question is, and I think the NIH, I was at a vaccine meeting recently where within the intramural program at NIH, they have some really promising candidates that they're developing and even thinking about moving into field trials pretty fast in Puerto Rico. But how it will actually play out in terms of a vaccine that's commercially available, I can't predict that time frame.
Great. Do our panelists have any additional comments or questions for each other? Sally, would you like?
I have a question for Ray, actually. So I work in congenital infections and treat, perinatal CMV infection, HSV infection, and we give antivirals to the infant, and that improves their developmental outcome and their hearing outcome. And so I was wondering if infants could be a target for your population to use the drug in, and it does require that we allow safety studies initially maybe in adults, but moving quickly into children.
Yeah, I mean, I think it's worth discussing. I mean, we haven't, as you mentioned, we haven't done any toxicology work in juvenile tox at all yet. I think we'd have to do that. Usually the pediatric work comes a little bit later after you get through the healthy volunteers and the adults. I think looking at what happens with the virus, particularly in the pregnant model and following the newborns and what happens there, I think would be very important to see that too. So we're definitely open. I think, again, it's what's happening with this virus, what's happening with the pregnant woman, what's happening with the placenta, what's happening with the fetus, all that, I think understanding that most certainly is something we could definitely evaluate, yeah.
Great.
Happy to work with you.
Good. Okay, well, please join me in thanking our first panelists. Okay, at this point, we're going to transition between the first and second panel. So if our second panelist could come join us, and then we also want to welcome Congressman Price back to the stage to moderate this second session.
All right, we'll move right ahead, and you can, of course, come and go as you will. But rather than take a break and have to reconvene, let's get going because we have limited time, and I think a lot of things to discuss. We're now moving to a panel of agency leaders, people whose responsibilities extend to this Zika crisis, the research aspects, the public health aspects, the kind of federal state coordination aspects. We have a panel well equipped, I think, to help us understand what's going on, what the priorities are that have been set, what the gaps are, what the needs are, what the prospects are looking ahead. So I'll briefly introduce our panelists. I will ask a couple of leading questions to get us started, but on this panel we want to move much more quickly to the audience participation. Hopefully we can use the previous panel as a point of reference, because we've gotten a very good overview, I believe, of the research priorities, some of the progress that's being made, but of course lots of questions raised about where we go from here. Left to right, or left to right from your point of view, on our panel, Dr. Richard Hatchett is the Acting Director of Biomedical Advanced Research and Development Authority at BARDA, part of the, of course, the U.S. Department of Health and Human Services. Next to him is Dr. Kelly Saldana, the Deputy Director of the Office of Health, Infectious Diseases and Nutrition at the U.S. Agency for International Development, USAID. Next to her, Dr. Cristina Cassetti, the Director of the Translational Science Program, the National Institute of Allergy and Infectious Diseases at NIH, and that is the lead institute in the effort at NIH, although by no means the only institute, as we'll understand when we turn to Kimberly Gray, who is the Health Scientist Administrator at the National Institute for Environmental Health Sciences, NIH, right here in RTP. And then finally, Randall Williams, Dr. Randall Williams, the Deputy Secretary of the NC Department of Health and Human Services. And then joining us online, I guess we don't have video, we're going to have an audio hookup, but joining us in a few minutes is going to be Dr. Michael Beach, the Associate Director at the Center for Emerging Zoonotic and Infectious Diseases at the Center for Disease Control, CDC. So that's our lineup, and we'll proceed with a few opening remarks, not formal presentations, and then quickly move to your questions, and we'll follow the same procedure we did with the previous panel. Let me begin with our two NIH representatives, maybe starting with Dr. Cassetti, and then moving to our NIEHS colleague. But Dr. Cassetti, if you could, of course, everyone expects NIH to be the tip of the spear in terms of medical research. This is, of course, a very complex research challenge, which involves many agencies, but we do look to NIH. I would say that the congressional appropriations that I was talking about earlier have very definitely mirrored this point of view, because in all the proposals that the administration has made, I think NIH is the only one that was not cut, and in fact was given more money than was asked for, which indicates a certain presupposition about where the thrust of the research is going to lie. I'm not asking you to comment on that. No agency would, no agency would ever reject additional appropriations. So I won't put you on the spot in that respect. But you could help us, I think, get started here with some reflections on the kind of research effort that you're overseeing. Whatever you want to say about it, but what you think we should know about the priorities, about how far it's gone, about the challenges ahead, just the lay of the land as you perceive it. And then we'll ask your NIEHS colleague to speak more specifically to the work they have underway.
Okay, thank you. So as you mentioned, NIAID is the lead on the research response to Zika at the NIH. And we are doing this in close collaboration with all our colleagues in different institutes, including the Child and Maternal Health Institute, NINDS, NIEHS, and NHLBI and others. And we're also collaborating closely with the CDC, FDA, and other federal agencies and BARDA to augment our response. We have 5 areas of priority for Zika research, and many of these areas have been already discussed in the first panel. So the first area is to understand the natural history of the infection in Zika endemic areas. So we're supporting several studies in different countries in Latin America to understand what happens when either a pregnant person or non-pregnant person gets infected with Zika. One of the largest studies that we have initiated in collaboration with EHS and other, and also the Fiocruz Institute in Brazil, is to study Zika in 10,000 pregnant women in different countries in Latin America to understand the correlation between infection in different stages of pregnancy and then the clinical outcome in the infants. And also what's special about this study is that we're going to be looking at the effect of asymptomatic infections and what happens to the infants, because most of the studies up to date have been done in symptomatically infected pregnant women. We're also supporting other studies in non-pregnant population to understand other clinical outcomes, including neurological outcomes like Guillain-Barré syndrome, as well as to understand what is the role of pre-existing immunity to other flaviviruses and the outcome to Zika infection, to see if there is any correlation between those two. The other area of priority is to very quickly jumpstart the vaccine development. There was some discussion in the previous panel. We have several candidates that we very rapidly initiated to work on. They were based on technologies that we already use for other flaviviruses at DNH. One is technology to make virus-like particles using DNA. This is from our Vaccine Research Center. We're using an inactivated whole viral particle, which is, we have a licensed vaccine for Japanese encephalitis based on the same technology. And we are doing this in collaboration with BARDA as well as Walter Reed. And we have also a live attenuated vaccine, which is building on our experience with developing live attenuated dengue vaccines. And also we have other candidates based on the nanoparticles that Arinda mentioned earlier, as well as a VSV recombinants, which were used in the response to the Ebola outbreak. Our third area is to improve diagnostics. As the previous panel mentioned, there's problems with cross-reactivity between Zika and other related viruses. So we're trying to develop the reagents and information to develop more specific diagnostics so you can diagnose after the fact, after the acute infection, and distinguish between Zika and other flavis. We're also supporting work on therapeutics, like the work that was mentioned before by BioCris and others. And also there were several efforts to improve, to understand the basic biology of the virus, the role of mosquito vectors, what happens to the evolution of the infection throughout the outbreak, and also importantly, to develop several new animal models which are supporting all the studies on vaccines and therapeutics.
All right, we'll come back to what other priorities you might view down the road. Let me just ask you quickly, though. How comprehensive a program is this, and how fleshed out a program is this? Is this, and how much of a crash program has there been in terms of making sure that that this was given a priority and that the word went out that proposals were not only sought but urged, and that this was not just an ordinary routine kind of program?
Yeah. So. This was put together very, very quickly after we realized that Zika was actually, in fact, a very important public health threat. And the way for us to move very quickly was to leverage our existing program on flaviviruses, which NIH has been supporting for many years, and to redirect these researchers to start addressing Zika using their expertise that was existing for other flaviviruses. And the quickest way to do that was to give them supplements to existing grants and contracts to very quickly redirect their efforts to address Zika. So we moved as rapidly as possible, but these are only beginning efforts. I mean, to sustain these efforts, obviously, additional resources would be very important.
Dr. Craig, could you chime in terms of the NIHS work that's underway that's relevant?
Sure. So as you know, we're not the institute that's looking at viruses, it's NIAID, but our lead is to look at environmental exposures. So the opportunity came as we were discussing what was going on at NIAID, that we already were funding a Superfund center in Puerto Rico that was looking at the effects of environmental exposures on pregnant women, and naturally we're also screening for viruses. And we were approached by NIAID and NICHD. Jose Cordero, who was the former head of birth defects unit at CDC and the former dean of the School of Public Health, is our lead in Puerto Rico on this birth cohort. So it was a natural partnership with NIAID and NICHD to have Jose's birth cohort and group and collaborative team under the direction of Akram Ashkabab from Northeastern to join the ZIP program, the Zika Infancy and Pregnancy. And it was pulled together very quickly. So I know they had an existing group that was already collaborating. It seems that the collaborators already knew each other pretty well. So it was a very firm partnership, and they've moved rapidly to put a protocol together, try to harmonize that and get that into human subjects review so that they can start. We would like to start in Puerto Rico very quickly. So we're hoping for July or August. And simultaneously, we all are working with our partners, our federal agency partners, and updating each other on activities. Last week in Puerto Rico, CDC was hosting a vector control meeting, and our agency, led by Aubrey Miller, our medical director, was with Jose there to talk about, we're trying to figure out what are all the pesticides and everyone's using in the home, outside, at not only in Puerto Rico, but we hope to find out what's going on in all the other pregnancy cohorts so we can kind of enumerate the threat of these pesticides. Because we have a lot of information, not only from our children's environmental health centers funded with EPA, showing that there are problems with pregnant women who use these chemicals during pregnancy and the short and long-term effects on neurodevelopment. So we're just thinking it's very important for us at NIHS to look at the potential impact that these pesticides may have, exacerbating an already important problem with the Zika disease. So the National Toxicology Program is trying to collect that data with CDC's assistance to kind of rapidly screen some of these chemicals to look at what the potential biological mechanism may be. Our office at NTP of OHAT, the Office of Health Assessment and Translation, is looking at systematic reviews. We're trying to find the big hits and what we need to enumerate. We have it in the protocols based by NICHD and NIAID to do the pesticide assessment, but we know it varies across countries. So that's our priority, to see how it may exacerbate the disease, how it may actually pass through the placenta, make the placenta a little bit more vulnerable. And we're really doing this with a community-based approach, because in Puerto Rico, there's so many Superfund sites. This is funded by Superfund. We already know there's already a burden on these pregnant women and children, and so we're trying to see the influence of that with the Zika, and that's where all our efforts are right now.
Dr. Hatchett, your agency's been referenced as obviously a collaborative effort with NIH, and I wonder if you could help us understand that. What is your piece of this puzzle? What kind of work do you have underway? What kind of path do you see going forward in terms of challenges that need to be addressed?
Sure. Thank you, Congressman Price and Dr. Holden. Thank you and to RTI for inviting us down to talk about what we're doing. If I could answer your question just by saying a little bit about what BARDA does, because I suspect
Sure.
that many people here in the audience. In fact, I see a lot of partners in the audience. For those of you who don't directly receive funding from BARDA currently, how many of you had even heard of BARDA before today? That's actually pretty good.
Pretty savvy audience. So.
Yeah, no, we were on a 60-minute special about Ebola, and we were called the agencies that no one had ever heard of. And really, given our mission, which is a very narrowly defined mission, you really don't want to hear about us. When you hear about us, it's a problem. Mm-hmm. What BARDA is functionally, I'll just take a second to describe what BARDA does. We support the development of medical countermeasures for public health threats that have national security implications, whether they are things that terrorists can use against us, things like pandemic influenza, or emerging diseases like Ebola and Zika. And by advanced development, I mean we support the development once a product moves into the clinical development space. And so we help companies perform clinical trials. We help them scale up their manufacturing capabilities. We help them with their regulatory efforts to achieve licensure or approval for their products. Now, with respect to Zika, like everybody else, Zika was not really on our radar screen before late last year. And we have a very simple strategy for responding to Zika. We are very much collaborating with our colleagues at CDC, at NIH. Within our space, we have prioritized 3 functional areas. We want to prevent Zika disease by supporting the development of vaccines. We want to enable the detection of Zika disease by supporting the development of diagnostics that could be used in commercial labs or in doctors' offices. Our colleagues at CDC, when Dr. Beach gets on, you'll hear about their efforts to expand public health laboratory capacity. We complement that by supporting the development of diagnostics by commercial firms. And then the last area that we have focused on is protecting the blood supply by supporting the development of screening tests for donated blood, as well as pathogen reduction technologies to take blood that's been donated and essentially to cleanse it of viruses, including Zika or other pathogens. So that's the main functional areas that we're working on right now, and we are doing that in close coordination with our partners across the interagency.
So presumably there has to be close coordination, especially with a program of this urgency, so that the division of labor is clear, the complementarity is clear.
So we have a virtual organization that has been set up for almost 10 years now called the Public Health Emergency Medical Countermeasures Enterprise. It's a mouthful, so we shorten that into an acronym like all bureaucrats do. It's called the FIMSI. And what the FIMSI is this virtual organization, essentially a roundtable where we gather with our partners who handle different parts of that product development life cycle. So with NIH doing the basic discovery work and the upstream development, the preclinical development, with our partners at the Department of Defense who do the same thing. We serve as a transition partner once they bring products to a certain stage of development, and then we carry it on, hopefully, to approval or licensure, and even, when appropriate, to stockpiling. And then we work closely with our colleagues at CDC who maintain the strategic national stockpile and think about how these products can be used to respond to public health crises. And critical to the function of the FIMSI as an organization and to its success as an organization is the role of the FDA in governing the regulatory process. And FDA is at the table with all of us through these development exercises, and that close coordination allows FDA to be very agile and flexible, which I would certainly compliment them as being in events like this.
Okay, is our CDC, Dr. Beach, on the line?
Yes, I am. Thank you.
Oh, good. All right, we're hearing you loud and clear, and then some. Would you prefer to wait a second, or have you picked up enough to? You fit right now. If you'd like to fill in your piece of the puzzle in terms of the research enterprise, the other aspects of CDC's work. Of course, you're an absolutely critical player in this. The level of the funding you're receiving has been a major issue in this, probably more with CDC than anyone else. But if you'd be willing, briefly, just to give us the short version of how your work intersects and complements what you've heard already from NIH and from BARDA.
Sure, that's nothing like jumping in right away here. Whoops, that sounds really loud. So I think what I'd like to do is just, first of all, we have a full court press situation here at CDC. We've had over 1,200 employees involved in the process. We've got deployments out to countries around the Americas, a very large contingent in Puerto Rico where the outbreak is ongoing, and then actually a very large component here in the continental U.S., Hawaii, and so on, planning for potential transmission in the U.S. Our areas, I think, as you've heard some from NIH and BARDA and that sort of thing, is, we have multiple facets that we go through for Zika. First of all, there's a large communications effort. What do you tell people? What guidance? What do you tell people on how to protect themselves? What do you tell clinicians from a clinical guidance and testing standpoint? We do investigations around cases to understand the disease itself. We're doing tracking of the disease and counting so that we can see kind of the extent and the spread of disease in different areas, particularly in Puerto Rico now. We're consulting on vector control issues. As I heard somebody say from the very beginning, dead mosquitoes don't spread Zika. So we would like to help them out in that case. In talking about vector control, we have a large component here, basically enrolling and tracking and following pregnant women in our pregnancy registries in Puerto Rico and the U.S. so that we can follow them, give guidance, testing, etc. We are involved with FDA, BARDA, and others in thinking about blood safety, both in Puerto Rico and the country. Again, some of our focus is on developing faster, quicker laboratory tests to diagnose Zika. Our main thing is to get testing out across these areas through our laboratory response network. So depending on what kind of spread of disease we see, we can pull in these laboratories across the country, mostly state public health laboratories, so that they can do surge capacity to make sure we can cover all people from a testing standpoint. We're conducting studies to learn more about Zika and its effect during pregnancy, both in Puerto Rico, Colombia, Panama, Brazil, and so on, to really understand what is, we're seeing the short-term impact here, but what's the long-term impact, both on pregnancy and birth defects, but also these rare conditions such as Guillain-Barré syndrome, which is a paralysis that occurs after the infection in rare circumstances. We're trying to understand how can we give guidance to people in this country and around the world about if you're infected, how long do you have to refrain from and think about protection during sexual activities. We know this is transmitted sexually well as working with people who travel around the world. And so understanding both from a male and a female standpoint what you need to be doing there to protect yourself, protect your partner, and so on is important as we update those guidance and follow people. This monitoring and reporting, I think, is critical to understand how the disease is spreading. We're providing guidance to travelers and Americans living in areas with current outbreaks, and we're really providing on-the-ground support for American Samoa, many countries around the Americas, Puerto Rico, and so on. We've got, I think these are critical areas. I think you've heard, sorry I had to come in late, my book with some other things, but this is a game changer. We have never had something like this in the past half century where we have this, where we've seen a virus that causes birth defects, where we have to think about really it's a game changer from a reproductive health standpoint, and now that women and their partners need to be thinking about how this can potentially impact them. And so understanding how we guide ourselves internationally, domestically, how we prepare for this domestically, I think is really critical. And we're working with state and local health departments now, including North Carolina, to think about how CDC put out funding to assist at the state and local level so that we can prepare for the potential spread in this country. We know that we already have over 600 cases of Zika infection in the country already. These are all travel associated so far. But we do expect, based on kind of our knowledge of other diseases like dengue and the chikungunya virus, that we will see transmission in the U.S. We don't think that will occur in the same way it is in Puerto Rico, Brazil, and Colombia, and that where it spreads through the whole country. We're a more temperate climate. The mosquito species aren't throughout the entire U.S. And what we've seen with these other diseases, dengue and chikungunya, is they tend to move in to the very southern tips of the country first and in through Hawaii, and then spread out a little bit from that. Most of it has been contained so far, but again, mosquitoes move. It's warm. Mosquito season's here, and so we're prepared to really take on this virus from a multi-state facet standpoint, moving up from the southern states as the season warms and as mosquitoes come out, to really prepare across the country for the potential for spread. We obviously are hoping not to see that, but I think we cannot afford to not prepare for it fully. I think that's probably more than you needed to hear, but I just wanted to give a short overview and hold you here for questions.
All right, Dr. Beach, thank you very much, and we will move along quickly. And just finally, we do want to hear from two others. The state labs, the state cooperative relationship has been referenced several times, and so we have Dr. Randall Williams here to briefly fill us in on this, and of course respond to questions, but give us the quick view right now.
Congressman Price, thank you, and again, on behalf of the department and the state, just incredibly appreciative to RTI for convening this group. One of the great privileges we have in North Carolina is the expertise we have. We've been around since 1877, and that's important because Secretary Brazier and I were down in Hosky in Hertford County on Friday, and that is apropos to this conversation because that county, Murfreesboro in Hertford County, produced the youngest graduate of the University of Virginia Medical School. He graduated at age 17 in 1869. Would anybody know who that was? He's still the youngest graduate ever from the University of Virginia Medical School. Dr. Walter Reed, whose wife, Emily, was from Murfreesboro, and where his home is still there. So we find that past is prologue, that 120 years later, we're still revisiting this. And so what we've done, the CDC made this issued a travel advisory on January 15th. We made it a reportable disease on February 1st. We had our first case in North Carolina on February 22nd. We've had 11 cases, all travel acquired. We've really concentrated our initial, because we had a little bit of window because it wasn't mosquito season, on pregnant women. I'm an obstetrician, so that's obviously something I'm very sensitive to. We have 175,000 women get pregnant in North Carolina every year. And so we're very proud that our state lab is one of the first that we can turn around the test. We can now, as of this morning, we can turn around the antibodies in 6 days. It was taking the CDC 6 weeks. If you can just for a moment take a chance and just sit there and think what it would be like to think that as a pregnant woman you've been told that you might have Zika and have to wait 6 weeks to find out what your antibody status was, and we've reduced that to 6 days. We're very proud of that, so we can let people know where they stand. We're working with our 1,700 obstetricians around the state in that, and working with our university partners very closely. As part of that, doing a lot of education, but now we're moving into mosquito season after Memorial Day. So now we're into prevention. And so we're in the process of hiring entomologists. We're incredibly appreciative of the governor and the Senate and the House. I'll be there in an hour over in the legislature as we educate them about the importance of the $750,000 that's before the Senate and the House in their budget to approve our entomologists and our state lab workers. And again, in the meantime, though, as someone mentioned, we're not resting on that. Thanks to the kindness of Michael in Western Carolina and East Carolina, we're already doing surveillance in 15 counties based on capacity and historically having a history sometime in the past of Aedes aegypti, and because they're kind of in metropolitan areas. So I'll stop there, but again, just incredibly appreciative to all the expertise in this room.
Thank you. And finally, Dr. Saldana, we don't think of USAID as primarily a research agency, and of course, that's only partly right. You do fund research. You, of course, fund lots of work in global health, and this is a global health issue. So can you give us a snapshot of USAID's activity to date and where you think this needs to go?
Sure. I'm really happy to be here. My first job in public health was actually for the Department of Health and Human Services in North Carolina, in the immunization branch, so I'm happy to be back. So USAID primarily works on implementation for Zika response internationally. To date, we're sort of beginning to move programs forward. We were able to identify, along with HHS, the reprogrammed resources that we were able to make available, but it took us a while to do that. We were actually unable to build on a lot of existing programs because we had begun 5 or 10 years ago a process of phasing out of many of our bilateral programs in Latin America and the Caribbean because they had reached high capacity and able to respond to the programs that we deal with. So going back in, we want to be very careful that we're not sending a message to governments that we have very carefully told over the period of several years that they have built up capacity and they can effectively, and they have strong systems to respond to programs, but to offer our help and assistance where we can. So in doing that, we are looking at supporting adding Zika diagnostics to clinical services, so ensuring that providers who are providing prenatal care services and antenatal care services, counseling services for pregnant women on reproductive health, that they are aware of Zika and know how to explain it to their patients, how to look for it. We're working with ministries of health on vector control, making sure that they're using the right kind of insecticides, that they have the right kinds of programs, that they're taking the right measures for this mosquito, and then providing some surge support at the community level, really where we think Zika will have a major impact. We are also working on the behavior change aspects and the communication aspects to make sure that populations understand the risk of Zika. There's lots of exposure in the region to dengue and chikungunya, but it hasn't really, the risks of Zika haven't really sort of permeated into the general consciousness in many countries. So not alarming people, but making sure they understand that there are real risks, that they need to take precautions, and giving them the tools to take those precautions, condoms for prevention, insect repellent, that sort of thing. We also have issued a grand challenge to identify, so it is research-based, to identify innovations to combat Zika and other future threats. So that's a major effort that we're also working in collaboration with colleagues. We have a really good partnership with CDC to do some of the broader work internationally that CDC has expertise in around the surveillance side and the laboratory capacity building, as well as close coordination around vector control and some of these other areas.
Good. I want to ask you a quick follow-up about Brazil, but why don't we gather people begin coming forward who want to ask questions, and we'll get our microphones ready to go. But there is a lot of attention, especially on Brazil, both because of the population, the degree of the threat, the Olympics coming up. It appears to be a dire situation. I don't know to what extent your agency is involved with that, but what would you say about Brazil in particular and their level, their ability to deal with this?
Actually, Brazil, we've found that Brazil has a pretty high level of ability to deal with it. They have been mounting a fairly effective response on their own. I know my colleagues up here probably have a better response, because really the collaboration in Brazil is more of a collaborative relationship and a sharing of efforts that I think the NIH and CDC are more involved in than we are at USAID, because it's not direct implementation. They have good capacity in that.
Any quick feedback on that?
I can just perhaps comment on the research expertise. Brazilians have a very long history of research on flaviviruses. Mm-hmm. So there is a very high level of expertise there. They've been studying flaviviruses, mostly dengue, for the last 50 years. So the response has been tremendous.
Good.
Yeah.
All right. Well, we have plenty of people lined up already, and just the same ground rules as before. Keep your questions as concise as possible, and address the specific members of the panel, and identify yourself as you ask your question.
One question I was told. Is it working? My name is Ram Ramabhadran, working in global health area. My question, oddly, is to Congressman Price. In terms of people, U.S. citizens, either athletes or spectators going to Brazil, is the government going to take a position either of not letting them go or testing every one of them who comes back?
I'm not the one to ask that. Although I'd be highly interested in the answer. I don't have an answer to that right now. I do know it's a matter of great anxiety and is being monitored extremely carefully. That's why I asked the question. Does anybody have a?
Probably Dr. Beach.
I'm going to say from CDC we do have a position on that.
All right, go ahead.
As you can imagine, basically we're not saying that people should not travel to Brazil. We're saying that pregnant women should consider not traveling to Brazil. But at this point in time, 40 million people travel by air through the Americas and that, and so we're not looking that we're going to be able to stop travel. This is really about thinking about pregnant women and protection, that they should consider not going during the Olympics. But not what we're saying is the same that we would say for any other traveler going to the Americas, is that they use precautions, repellents, and so on that we put out there for all travelers.
All right, thank you. I understand our News and Observer reporter has a question.
Thank you.
All right.
My name's Steve Ginley. Like Representative Price said, I'm from the Raleigh News and Observer. Michael Reiskind mentioned household measures as being really effective against Aegyptus, especially the mosquito. I was wondering if, Michael, if you could speak to what household measures the CDC recommends. Maybe Dr. Gray as well might have some comments on that.
I'm going to defer.
Did you hear the question, Dr. Beach?
Yeah. Yes, I did. And so you're talking about false reduction in household measures? Yeah, sorry, there's a real echo on my line.
Go ahead. He's asking you a question.
Yeah, mosquito control as a household measure, as well as larval sites.
Mosquito control as a household measure, larval sites.
Yeah, okay. I mean, I think what we want people to do is once a week, walk around, empty and scrub, turn over, cover, look for those areas that are accumulating water, tires, buckets, planters. You know how we have things laying around outside the house and that sort of thing, bird baths, flower pots, anything that's got water in it can potentially be used as an egg-laying site by Aedes. And so by the Aedes aegypti or albopictus, the two species that we think are the ones we need to worry about here. So tightly cover any water storage containers. If you're collecting water outside, make sure it's covered with a screen that's small enough to keep mosquitoes out. The larvicides, that they kill the little larvae that are in the water, and that you can buy those at some of the big box stores and put them in areas where you think that you're going to have mosquito larvae occurring. You can use sprays and that sort of thing, but I think make sure that you're using them according to the manufacturer's areas. But remember that Aedes aegypti is essentially the cockroach of mosquito species. The problem with it is that it's an urban mosquito. It tends to like to live where humans are living, and so it's in the house. And it bites during the day. It's not the type, these nuisance mosquitoes that can be out there, at dawn or dusk. It tends to be biting during the day. It'll be in the humid areas of the house, under sinks, in closets, in the laundry area, and that sort of thing. And so you do want to make sure that you're looking at, are your screens intact? Have you dumped all these water areas? If you have a septic tank, you don't have cracks or gaps in it, that sort of thing. You want to cover openings and vents and plumbing pipes. But you really want to kind of make sure they're not getting into your house easily, because that's where they're going to bite during the day, and they bite multiple people with the same bite. And you want to get rid of all of the water areas that you can see around the house, because they tend to be, unlike other mosquitoes, they're a bit tough in that they lay eggs in multiple areas. So doors, screens, don't leave your door propped open. So that you allow them in. Air conditioning. One of the reasons we think that the spread in the U.S. of some of these other viruses is not as wide is that we have air conditioning and screens, and so use them. And then, you can use some insect sprays and that sort of thing in your house if that's what you want to do. But just really be careful about reading the precautions and don't overdo it. Those pesticides they're labeled for safe use if you use them the way that you're told to do that.
All right, thank you. And here we go.
My name is Hannah Hodge, and I'm a student at the University of North Carolina at Chapel Hill, working with FHI 360. And it's evident that there's been a very robust national and international response to Zika virus research. But one of the things that was mentioned earlier is that this virus is distinguished by its sexual transmission and the threats to fetus and maternal health. So I'm wondering if anyone can speak as to what the national and international focuses on family planning are going to be moving forward, and thinking about sort of the very alarmingly high rates of unmet need, especially in Latin America and in the US, and unintended pregnancy rates at a time when those are more harmful to public health than ever, what the response to that is going to be from the U.S. government.
Sedona?
Sure. I can talk the international side. So I mentioned earlier, we have phased out of much of Latin America. We still have ongoing programs in Haiti and Guatemala where we do work on improving access to contraceptives and reproductive health. We, in the rest of the region, we very deliberately graduated countries from family planning assistance from USAID, so we are very careful about going back in. That being said, we do have some sort of legacy systems that were set up. We, in many countries, set up what's called contraceptive security committees, which bring together all the key stakeholders in the country from the public sector, social security sector, private providers, to sort of make sure and sort of take a look, is everything functioning correctly with international tenders for contraceptives? Are there stockouts in certain places? And they can meet together and sort of jointly come up with a way to identify and solve problems. So we're looking at sort of revitalizing those committees and making sure that they're functioning as best they can to increase access. We also do have, as I mentioned, a strong component to train providers on counseling, making sure that they're able to communicate the risks to pregnant women so that they understand, and women of reproductive age who may not be pregnant or may not be thinking of becoming pregnant, so that they understand and have access to contraceptives if they choose to delay their pregnancy. So we're working both on a provider side and an information side, and then also within the system.
Any other comments on that? All right.
This is Michael. I just say that this is basically access to contraception is one of the three things that we think are critical from a protection standpoint. Vector control, protecting pregnant women from mosquito bites, and access to contraception, I think, are the three pillars of how we can protect people.
All right. Next.
Michael Gunn, Duke University. I work in the area of diagnostics. The NIH has been very responsive thus far, and our program officers have been excellent. Historically, you get very quickly to a point with diagnostics where you need to validate your test, whatever it is, against human samples. In the case of Zika, specificity is the huge issue. So individual researchers need access to samples, not only from Zika, dengue, yellow fever, everything, in order to validate our assays. Is there any mechanism in place or any planned mechanism by which individual investigators could obtain such resources without having to contact everyone across the planet?
Maybe I'll start and
Sure.
Richard can comment. So NIAID is supporting a large reagent repository, the BI, and we have been very actively trying to stock our repository with serum from Zika patients after acute samples and after recovery. So we have definitely some of these reagents available for distribution to the research community. We don't have enough, so we are still trying to look for additional reagents. In fact, some of these reagents came from researchers from this area. They were very kindly donated serum to us. But we're also in very close collaboration with the BARDA that are putting together a large panel of such reagent serum. And maybe Richard can comment.
Sure. One of the services that we provide our product development partners, which are companies, not individual researchers, and I know that's the source of your question. We have a clinical studies network, and we have contracted with one of the clinical research organizations in our clinical studies network to collect particularly convalescent samples, primarily to put together formal validation panels to assist diagnostics companies with validating their tests. We have not extended that to individual researchers yet, but certainly we are working closely with NIH. Access to samples and specimens has really proved surprisingly challenging for everyone, not just for the individual researcher.
Randall Williams was about to chime in on the very practical question that our reporter raised, and so let me give you a chance to do that.
Thank you so much, Congressman Price. It's a great question. If you'll go to the website www.ncpublichealth.com, it gives a list, because that question is going to come to the forefront very shortly. What can I do around my house? And it'll give you a descriptive list. So thank you very much for the question.
Okay.
Derek Nimmo from Oxitec. My question is related to the first question as well. I've worked around the world with different mosquito control agencies from Malaysia, Brazil, Panama, Cayman, Florida. And to reflect on Dr. Beach's comments, Aedes aegypti is very hard to control. I'll give you an example. The Florida Keys Mosquito Control District, they spend $1.1 million every year just on Aedes aegypti control. They have 3 helicopters, they're using BTI, they're using insecticides, they've got 30 staff going around doing door-to-door, putting on door knockers, giving out information, and basically trying to control this mosquito. They can only reduce it by 30 to 50% at best. And they had an outbreak of dengue in 2009, 2010. They've been saying since then they can't control this mosquito enough. And in the past 2 weeks, they've had a dengue, locally transmitted dengue case in Key West again. So to me, it looks like current control measures just don't work against this particular mosquito. So are you looking into new technologies? Are you thinking about them? Because from that point of view, I can't see what advice you'd give to people in Caribbean, Puerto Rico, Hawaii, Brazil, to actually control this mosquito if the best in the world here can't control it. Bit of an open question, really, but basically, you know, trying to challenge you on sort of what you're trying to say to people and how you're trying to say it, and with that premise that we can't control this mosquito with current technologies.
Yeah, to speak to North Carolina, and I think it was alluded to earlier, we think that one of the advantages North Carolina has is that to have an epidemic, you would need a critical mass of mosquitoes and a critical mass of people, as you see in the South American countries, where it is a confluence of factors, people being outside, not inside, much more of the Aedes aegypti mosquito than our indications would show we have here. So we're very appreciative of that. But to your point, in those countries in which you don't have, you're not that fortunate, where you have a lot of the mosquitoes, you have a lot of people outside, I would have to defer to others, but I would agree with you. And then you have a mosquito that for the first time creates a vector-borne birth defect. It is incredibly challenging.
So on the
and from a CDC standpoint,
go ahead, Michael.
Please, go ahead.
I think from a CDC standpoint, we would like to see these technologies looked at. We'd like to see the approvals, if everything passes muster, that they're done so that we can evaluate them more closely, because I think they have great promise, you know, in Florida Keys, Puerto Rico, and other areas that the approval process.
And on the international side, I mentioned that we issued a call for innovative ideas around Zika and other future threats. One of the areas that we expressed interest in looking for was around vector control. The applications for the Zika specifically focused innovations have closed, but the future threats one, which is a little bit more expansive, is open until June 17th. We've got people reviewing those applications now, and we hope to identify some innovative solutions sort of across not only vector control, surveillance, diagnostics, community mobilization, behavior change that have promised to be quickly introduced. So the idea is that we want to start in these countries with what we know. We want to sort of first look at what are they doing right, where can they be improved upon with existing knowledge, and then as new things are identified and can be brought into the mainstream, we also have platforms there to be able to quickly introduce them and scale them up. So that's sort of the plan.
All right, here we go.
Hi, my name is Andrew Kraska, and my question is to possibly Dr. Williams and Dr. Cassetti. As you mentioned before, there's around 600 cases in the United States right now, all travel-related. Are there plans in place where if we do reach that critical mass in some areas, say the Keys, that we will actually be able to put people there to try to control that rather than the work that we're doing internationally to try to help?
NIH doesn't, this is not exactly our area, is to control. The outbreak response is more for the CDC, I would say.
I mean,
this is Michael from CDC.
Michael, if you can respond, but let me ask you, Michael, hold the phone a bit away from your face if you will. You're breaking up on us.
Yeah, no, it's actually.
All right, go ahead.
Yes, we do have a very vigorous response from the state and local health departments around the country. We have CDC has a group of
Sorry, Michael. It's incomprehensible now, I'm afraid.
Okay.
Maybe just
let's go ahead. Hear someone else, then we'll pick up on you in a minute. Hopefully, it'll be better.
Perhaps to help Michael, you asked about outbreaks in the United States. We have a very significant outbreak in Puerto Rico, and so we are actively responding in Puerto Rico, and don't want to forget that the territories are certainly part of the United States. And Michael, if he's able to figure the phone out, can probably address the response there, which is a live fire exercise. Not even an exercise. It's a full-on response right now.
And I would just add, I think that Michael and I are in very close contact. I think across the government, we actually have pretty close coordination, and so just like on the international side, we want to learn from what CDC and HHS are doing in Puerto Rico, then we would hope that if we develop things that are working on the international side, that those can be taken to learn domestically, and we're sort of sharing across.
Yeah, I just wanted to add, with the Zika infant pregnancy study, part of that is in Puerto Rico. So we're going to see the etiology of the Zika disease in early pregnancy. We're talking before 12 weeks of pregnancy. So that's going to be informative with CDC efforts and surveillance and what messaging we're going to have and what is the critical window of exposure. So don't think this is just for Latin America, but the data of the 10,000 pregnancies will be so valuable as we move forward, as we describe and characterize what the spectrum of disease will be. Besides everyone just focusing on microcephaly, there could be actually some other soft neurological signs as well that we need to consider. So it is a valuable opportunity for us to get ahead of what's upcoming, maybe.
Michael, you want to try again?
Yeah. How's that?
Better.
Okay. Thanks. I think it is right. We've talked a lot about international things, but we work hand in hand. Kelly and I, USAID, are hand in hand. We work for NIH, Florida, FDA, all of the other organizations at the U.S. government level are all coordinated in developing a national response. That's through emergency response teams, facilitate funding to basically expand the laboratory.
Sorry, Michael, I'm going to interrupt you again. It's just not working. Maybe you can get on another phone line. We'll try to help from here if some of our folks can work on this. Sorry about that. Okay, next question.
Hi, I'm Bill Pan at Duke University. I just came back from the Amazon yesterday, and I got 3 emails about this meeting, so I'm glad I came. So I have a question for Dr. Gray about the Puerto Rico study.
Sure.
So I had a couple questions. Number 1, I'm really curious to know what pesticides you're looking at. Number 2, I'm wondering if you're looking at other chemical exposures like metals, arsenic, other things like that. And then the third question is really, I guess it's open to the panel about whether or not you're looking for other types of exposures like climate change, land use, interactions, or something like that.
So let me start by saying that PROTECT, under the birth cohort under the guidance of Jose Cordero, is funded under Superfund program. So that started over 4 years ago. They've already recruited 700 pregnant women, and what we're doing as part of the ZIP study is enhancing that program to enroll in 5 other regions and to increase the number of pregnant women that they can enroll. And then as part of the children's study, they're following them up over time and looking at neurodevelopment and other effects. Puerto Rico, as you know, has like 12 Superfund sites. They have multiple exposures, a high rate of preterm delivery in that territory. So we have been, through Superfund, been characterizing the exposures of the pregnant women in that community for the last 4 years. So yes, metals, arsenic, phthalates are a big contributor in that area. They're also looking at air quality from some of the distribution from some of the manufacturers. So we have. And then what's really interesting is we took the protocol from Protect, and Jose gave it to NIAID and NICHD, and all the other countries have used that protocol as the standard to try to characterize other exposures in that community. So it's not just environmental, there's also the social aspect, the stress aspect, and we're trying to get a handle on even the household environment. We're trying to characterize that as well. So this is an effort across multiple regions, multiple cohorts, and we hope that the data will be useful. It should be useful. The pesticides that we're gonna do a screen of pesticides because each region is different. So we couldn't come up with a one-size-fits-all, but we will have access to these biological samples that we're banking for pregnant women. It's every 2 weeks of urine, every month of blood. It's even more if you're Zika positive. So we will have a lot of biological samples, and as you know, some of these environmental contaminants that we measure are kind of difficult to measure, so we may have to use some broad category like a DAP, dialkyl phosphates in urine, which will be very easy for us to do, and we hope to leverage, and we're still in the beginning process of talking who's going to analyze what, but we have a cheer laboratories across the United States. One of them happens to be funded to that Superfund site of Michigan, John Meeker, who runs that project in Protect. So they will have the abilities and facilities to actually characterize those biological samples for environmental exposures for all of the countries that are able to give us their specimens.
All right, let me ask a question that's, I'm not asking here for political commentary, but I am asking for, if you, to the extent you can back off a bit and talk about what you've undertaken thus far, what you're projecting and planning, and to what extent, where the next dollars will be spent, and what depends on those dollars being there. I mean, we stipulate that, and we can pick this up from the researchers from the first panel, people have been moving money all over the place and switching priorities and putting an effort into this with their own resources, and you know, that's what the agencies have done as well. We know in particular how much money has been moved around from the Ebola effort and from state public health preparedness funds. So that is going on, and that's what accounts, I think, for a lot of the work we've heard about here today. However, we, with the exception of NIH, but you can chime in if you wish about the funding picture there, but with the exception of NIH, we're talking about the global health aspect of this. The funding, slated funding, is less than 1/3 of what was requested. BARDA, what is the BARDA figure?
The BARDA request is for $188 million, and we actually, like our colleagues at NIH, actually do reasonably well under both of the current bills, but in the absence of a supplemental, it would very significantly impact our ability to proceed with vaccine research in particular.
Yeah, the question is partly the amount, but also partly the timeliness of the bills being passed, which have to do with the current fiscal year, but also the next fiscal year.
So just to address your question, with the reprogrammed funds, we have a number of negotiations ongoing with potential partners for diagnostics, for vaccine early stage vaccine development, and for protecting the blood supply. And the reprogrammed funds from Ebola, which I will say I agree with the sentiment that taking money from Ebola to address the Zika problem is very unwise because we haven't finished the job with Ebola. But with that money that has been reprogrammed, we anticipate executing most of that probably over the next 3 to 6 weeks. And then beyond that, the absence of funding would very significantly begin to limit what we can do.
All right. Michael, we're going to try again with you in a minute, but I want to ask our USAID person to comment first.
Sure. Okay. So I believe the supplemental request for USAID is $325 million. I think under the House bill we get $100 million. Under the Senate we get $225, something like that. So either way, it's less than we're able to implement. to implement fully the picture in covering enough countries to really have an impact on interrupting transmission and the potential of transmission to come to the US, we really feel like we would need to cover a large portion of the region. Right now, we've got 5 countries that we're looking at focusing on. If we don't have additional money, we may be limited to those 5, and then within those, our activities are more limited to the areas that we can target as the highest risk.
Michael, can you chime in? CDC is, of course, the main agency that's talked about in this connection.
Okay, yes, and hopefully the echo is gone now.
It's better.
Uh-oh. Oh, it is better, okay. So, again, I think out of the administration request for $1.9 billion, 743 was for CDC, and we talked about some of those activities. What we're running on right now, basically, is AID has reprogrammed some Ebola money, and we're doing that internationally. We've reprogrammed money as well for Ebola money, and we're using other funds that are preparedness related. But I think it is one of those issues where we're robbing Peter to pay Paul sort of thing, is that pulling funds out of Ebola areas and global health security is really detrimental to those programs, and long-term protecting health globally is how we protect ourselves here as well. So I think we've started up multi-year studies. Those are going to be extremely difficult to continue without additional funding. As you've heard, these long-term studies on the impact of these birth defects, I think, are incredibly important to understand what's going on, and that's very difficult to continue those in future years. Helping states track and control mosquitoes, that's part of what we fund. We fund laboratory, to the state. So vector control, laboratory, basic kind of surveillance and tracking of disease, those are all coming out of our preparedness and our epidemiology and laboratory capacity grants, which those are, we need the full supplemental to really make the full impact so that you in North Carolina and other states can be fully prepared for this. So it will be, basically where the next dollars will be spent, we're not sure because we're essentially using up everything that we have at this point in time.
All
I
right.
hope that addresses your question.
Yes, it does. It sure does. Dr. Williams, what's the state funding situation, and to what extent, I'm not sure most of us, I certainly don't know exactly how this works, to what extent you're passing through federal funds, to what extent this is a state appropriation, where does that stand?
We just received $400,000 that we're pushing out to our 100 counties for education. We've been incredibly appreciative.
Receive that from where?
From the federal government. I can't remember which grant it came from, but that goes out in July. And then we have been incredibly appreciative of, as Michael was saying, the work with the state lab. The fact that we've been able to get our test turned around to 6 days has certainly been a large part due to their efforts. And then on April 1st, we were down at the CDC with Dr. Frieden, 21 other states, and that was incredibly helpful to see what our peers are doing. So again, our modus operandi is always to partner, and so we greatly appreciate the partnership from the federal government.
Can I just
Yes.
make one statement? So
Sure.
NIH, we have a Zika time-sensitive funding opportunity right now. So it's small grant awards. For those of you at the universities know, these small grants are $275,000, 2 years. But the interesting thing, if you have an interesting idea that's responsive to this opportunity, it's quickly received, quickly reviewed, and funded, with cutting the time probably by 2/3. So NIH has a global call for applications. NIEHS is just using its usual time sensitive. We've always used it for wildfires, environmental contaminants, but if you're looking at environmental chemicals and Zika disease and transmission and exacerbation, we also would welcome an application as well, and that's on the NIH website.
All right. Good. Any further questions from the audience? Yes. Oh, our researchers. Yes, we should give you a chance, please. See if we can get a microphone to you.
So I just want to make a comment. I think we've heard a lot of ideas about controlling Zika in the US. Controlling Zika in the US, and I think a lot of the things that we talked about, surveillance, identifying cases early, and I think those would be very effective because we are not dealing with a situation that's highly conducive for transmission. But I also want to remind everyone that globally, what we've seen with dengue over the last 20 to 30 years, it's an enormous amount of resources gone into vector control, and it has had no impact. Dengue, really, globally, dengue transmission really has been a global public health failure, right? Every decade, the virus continues to expand. There's more and more cases, and really the efforts at controlling the mosquito have had very little impact. And so why is that? It's not because insecticides don't kill mosquitoes. It's because the larger societal changes, right, are outstripping anything that the vector control people are throwing at the mosquito. The massive urbanization that's taking place, rural to urban migration, and we are creating conditions for the mosquito, favorable conditions for the mosquito, much faster than the controls that we are implementing. So globally, looking at Zika, I think this virus is going to be around for a long time, and it's not as if we only tried to control dengue a few years ago. It's been going on for 30 or 40 years, and given the ecology of Aedes aegypti. The other thing I really want to make, this is especially for CDC and USAID, is talking about the Zika pandemic now in Latin America. The ecology of Zika is similar to dengue, and less than 10% of the world's dengue cases are in Latin America. The vast majority are in Asia. And we really need to be thinking about what happens when if this virus gets into Asia, and the global scale of this problem. In countries like 300 to 400 million dengue infections are documented every year. And if Zika starts to do something similar to what it's doing now in Latin America and Asia, the scale of the problem is massive.
Well, would one expect that? I mean, what is it possible to predict?
There's no reason to think it wouldn't, because given the transmission cycle, Aedes aegypti, susceptible people, there's no reason to think that Zika may not emerge. Now, there's some things that we don't understand. Zika has been around in Asia, but it hasn't caused large epidemics or severe disease cases like what we're seeing in Latin America. One possibility is that it's a new strain. And what happens if this strain goes back to Asia? Another possibility is there's something that we don't understand in the Asian setting that in some way doesn't allow Zika to emerge, and I can't think of what that might be.
So Michael may want to comment, but I know CDC is looking into those questions. There are places where Zika is endemic, has been around for a while, and we haven't seen the microcephaly. And so is it a new strain? Is it poor surveillance? We haven't been looking. Is it something else? But CDC is planning to look into that.
Michael, you want to comment?
Yeah, I think it's a really important question that was brought up, that we are looking and we'll be looking in some of these what we call endemic countries. They haven't seen epidemic. We're not sure that they didn't at some point, but clearly we want to understand how does the disease behave in those countries, and what percentage of microcephaly is actually due to Zika. No one's really looking, and so that will be part of what we're asking. So we understand where many of the epidemic countries now may end up having disease more like these other countries. So we want to understand how that transition occurs. I will point out that again, the Aedes aegypti, there were many programs in place decades ago, and essentially we defunded them. And we've seen the result of defunding many of those programs is that Aedes aegypti moved back into the areas where it was under better control than it is now. Thank you.
The defunding was with respect to domestic or international control efforts?
Many international programs were kind of dismantled.
Mm-hmm. All right, looks like we have one more question. So,
it's more of a comment to actually Dr. De Silva. Richard Reitinger, I'm from RTI and I lead the Global Health Division there. I think I agree with you that vector control with regards to dengue has been complex and certainly could have been more effective and more successful, and there are reasons why that is so. But I think what we've also seen for other vector-borne diseases, and specifically malaria, is that global initiatives, when really well coordinated and when there is the political will and commitment, can be hugely successful. I mean, when you see the malaria landscape, bed nets and IRS and other vector control interventions have been dramatically reducing the burden of malaria globally. And I think for dengue, there has been a lot of investment, but it hasn't necessarily been at global level, and it hasn't necessarily been coordinated to a global extent that, for example, it has been for malaria. So there are certain vector control approaches that have shown some promise. I mean, we talked a little bit about Wolbachia and the genetically modified mosquitoes. In Australia, they have done some targeted IRS that has resulted in actually some very good impact on dengue there. And that's why I wouldn't say necessarily vector control, may not be a promising approach, because also, as we discussed earlier on, I mean, the therapeutics and the vaccines are still some time away to actually being operationally scalable to the extent that we might want them to be in terms of the Zika response.
Yeah, I think my point is, I think the urbanization globally, the level of urbanization that's taking place, it's different from malaria. That uncontrolled urbanization, that's what's driving the epidemic. And we will be able to control vectors, but it's going to be hard to keep up with the extent of urbanization.
Any parting shots from our panel?
Just one other thing I wanted to mention is I think where our funding is focused on implementation in Latin America, we are working closely with WHO and UNICEF and enabling them to be able to respond as the need arises in other countries. And then if we had additional resources and see the need globally, we're also prepared to respond. And then feeding into sort of the what CDC learns through what they're looking at.
All right, I'm going to turn the podium back over to our RTI hosts, and I'll do that with a hearty word of thanks for putting this together, helping us in so many ways to assemble two outstanding panels. And we appreciate everyone who's made the effort to be here. Thank you, Michael, also for joining us remotely. Thanks to everyone who's here. We will look forward to staying in touch. I hope that this has been a helpful session, though, in terms of informing us better about what each other are doing and about the requisites of success going forward. This is a big one, and it's a big one politically as well as in other ways, and we have a lot of work to do. Thank you so much. Thank you to RTI.
And please join me in thanking Congressman Price for this idea and the inspiration. Okay, just three final announcements and invitations. For those of you that are interested, we will have the videotape proceedings available online. If you can go a little bit later this week to rti.org/Zika, you'll find the proceedings there. We do want to encourage, for those of you that had questions or comments that didn't quite make it into this discussion, the discussion will continue online, and so do join us via social media using the hashtags that we talked about earlier, RTI Forum or Zika NC. And last, but certainly not least, and probably important to many of you, we want to invite everybody to lunch here, and so you'll find that in the lobby. So thank you very much for your time and attention today. Thank you for taking the time to make it here to RTP. Thank you.